So next we will have Natàlia Feiner and Suris that will give a talk entitled Nanomedicine, New Solutions for Neurodegenerative Diseases.
Hello. Hello. Good morning, everyone. Good morning. We are Natàlia Feiner and Suris, and we are going to present Nanomedicine New Solutions for Neurodegenerative Diseases. This work was carried out in the Institute of Advanced Chemistry of Catalonia from CSIC.
In the last decades, it has been an increase in life expectancy, as it can be seen in the upper graph. Also, it has been an increment in the prevalence and the incidence of neurodegenerative disease. It can be appreciated in the lower graph that the places where are more death caused by Parkinson's disease are those with higher life expectancy. And we can also see that it has been an increase of the number of new Alzheimer's cases detected each year. Also, nowadays, there is a current lack of an effective treatment, mainly due to the blood-brain barrier. And for this reason, this causes huge social and economical cost. So we have to try to trespass this.
So what's the blood-brain barrier? The blood-brain barrier is a physiological barrier that separates the blood from the brain, acting as a natural membrane. So only certain type of molecules can pass through it. Therefore, novel diagnostic and therapeutic agents are required. In this context, nanocarriers can overcome these limitations due to their own characteristics.
Nanocarriers are transporters of drugs and other molecules with size between 15 and 500 nanometers. You can see on a scale of it there. These nanocarriers can be made of different materials, such as organic materials like liposomes, polymeric nanocarriers, or chair protein basics, or also of inorganics like carbon nanotubes or magnetic nanoparticles. They can also have different structures and characteristics. In our lab, we work especially with polymeric nanocarriers, with polymeric nanoparticles made of biocompatible and biodegradable materials. That means that they can be introduced into the body, and after a certain of time, they are degraded and eliminated without causing any damage. They are also enable a sustained release of the drugs and reduce the side effects. There are different methodologies to produce this kind of nanoparticles, such as salting out, nanoprecipitation, or solvent evaporation. In our case, we work with solvent evaporation when we take nanomulsions and we use as a templates.
Okay, so nanomulsions are fine emulsions with sizes between 20 and 200 nanometers that show some interesting properties due to their nanometric size. The most interesting properties are their stability against sedimentation and creaming, and also their transparency or translucent aspect. We can form oil in water nanomulsions, in which the oil is the dispersed phase and water is the continuous phase. And they can be stabilized, this interface can be stabilized using surfactant molecules. Among all the fabrication techniques, we focused on the phase inversion methods, specifically in the PIC method, which is a change in the composition of the nanomulsion at constant temperature. Nanomulsions can have a wide range of applications, comprehending formulations, reaction media, or extraction media, but we use it as a template for nanoparticle fabrication.
As my mate has said, we produce nanomulsions using the phase inversion composition method. It consists of the stepwise addition of water into a mixture of oil and surfactant. With that, we first have structures of water in oil, then they pass through a bicontinuous phase, and finally we get the water in oil nanomulsions. These nanomulsions will be used as a template to form the nanoparticles by evaporating the solvent in reduced pressures.
So these nanoparticles have very wide range of applications as well. The main general applications in the biomedical field are from therapeutics to the diagnostic purposes. They can act as advanced delivery systems for drugs and as well as non-viral gene delivery systems. And for diagnosis purposes, they can act as magnetic resonance imaging agents and fluorescent tracking. These applications can be achieved by encapsulation of desired molecules inside or in the surroundings of the nanoparticle, and also with surface functionalization.
What we do is encapsulate different kinds of pharmaceutical actives. With that, we want to reduce the frequency of drug administration, also enhance the drug efficiency, and finally, reduce the side effects. We will also encapsulate fluorescent compounds to use them as a medical diagnosis systems and to track and target a specific organ. Finally, we can attach some specific molecules to the surface of this nanoparticle. We want to attach cell-penetrating peptides that are a kind of small proteins that can, by their own, penetrate the cell. So with that, we want that the nanoparticles can enter to the cell and act there.
So we will give you a glance of our results. Once we fabricated the nanoparticles from nanomulsion templating, we can observe that they have a really small size. Nanoparticles without any by itself are from 25 nanometers of radius, and they are very stable. And we characterize also nanoparticles with an encapsulated fluorescent dye and with some drugs for neurodegenerative treatment. And also, we functionalize their surface using cell-penetrating peptides. We obtain nanoparticles with successfully encapsulated fluorescent dye. As we can see in the image, nanoparticles with the fluorescent dye cannot pass through a membrane, whereas the fluorescent dye by itself in an aqueous solution can pass through this membrane, so the encapsulation was achieved. And finally, we also looked for the toxicity of these particles, because if we wanted to use it as for biomedical purposes, the toxicity has to be looked. As we can see on the plot, fluorescent and drug-loaded particles and functionalized particles had the same survival expectancy on cells than those of the negative control.
To conclude, say that nanoparticles has been successfully obtained from nanomulsion by the low energy methods, following by a solvent evaporation. Nanoparticles are able to encapsulate different kinds of molecules, maintaining their stability and size. And finally, nanoparticles are promising systems to cross the blood-brain barrier due to their small size, functionalization, and no toxicity, thus being appropriate nanocarriers for treatment of the neurodegenerative disease. Thank you for your attention.